Hey there, and welcome. Today, we're going to take a deep dive into the microscopic world to unmask a really common, and let's be honest, often super painful intruder. You know the feeling, right? Waking up and it feels like you're swallowing glass. The fever, the chills, you just feel miserable. So, what exactly is the culprit behind all that trouble? Well, let's meet our prime suspect. It's a bacterium called Streptococcus. For this explainer, we're basically going to open up the case file on this bug to figure out what it is, the kind of damage it does, and most importantly, how we fight back. All right then, first things first, let's get properly introduced to the bug itself. So if you were to look at this thing under a microscope, you'd notice a few key tells. Scientists call it gram-positive, which is just part of how they classify bacteria. It's spherical, so it looks like a little ball, and it has this signature look of arranging itself in chains, almost like a tiny string of pearls. So how do scientists tell one type of strep from another? I mean, there are lots of them. Well, it turns out they have a really clever method. It's all about watching how the bacteria acts when it's introduced to a plate of blood. And that brings us to this key process called hemolysis. You can think of it as the bacteria's signature move. By watching how or even if it destroys red blood cells, we can build a really clear profile of our suspect. And there are basically three main ways this can go down. First up, you've got alpha hemolysis. This is where the bacteria only partially breaks down the red blood cells. What you see is this kind of murky, greenish glow around the colony. It's a clear sign of partial damage, but not total destruction. Next, we have beta hemolysis. Now, this one is the big one. This reaction is way more dramatic. The bacteria completely decimates the red blood cells, leaving a totally clear, colorless zone around it. This complete clearing is a major clue for identifying some of the more dangerous strains of strep. And finally, there's gamma hemolysis, and this one is super straightforward because it means nothing happens. The bacteria grows just fine, but it leaves the red blood cells completely alone. No destruction, no color change, nothing. Okay, so we know how to spot strep in a lab, but what kind of trouble does it actually cause inside the human body? Well, it turns out this bacterium has a lot of different faces. The illnesses it causes usually fall into two main categories. First, you have the pyogenic infections. These are the direct, on-the-spot, pus-forming infections right where the bacteria gets in. And then, you've got the non-suppurative complications, which are more serious conditions that can actually show up after the initial infection is gone. So let's look at those direct infections first. We all know the classic sore throat, what doctors call pharyngitis. But strep can also cause scarlet fever. It can infect the skin and wounds or burns, or lead to conditions like impetigo. And in really severe cases, it can escalate to something called streptococcal toxic shock syndrome. And that is incredibly serious. We're talking about a situation where entire organ systems can start to shut down. But the story doesn't always end there. And this is exactly why getting treatment is so important. A strep infection can trigger these really serious secondary problems. For instance, acute rheumatic fever, which can follow a sore throat, can cause permanent damage to your heart. This happens because your own immune system gets, well, confused. It's trying to fight the strep, but it accidentally starts attacking your heart tissue instead. Then there's acute glomerulonephritis, which can happen after a skin infection and do serious damage to the kidneys. So, you feel sick, you drag yourself to the doctor, but how do they know for sure that it's Streptococcus causing all this misery? Well, let's follow that sample and go inside the diagnostic lab to see the detective work in action. It all starts with that uncomfortable throat swab, right? That little sample gets sent off to a lab, and that's where the real investigation begins. So what happens next? Well, the process is pretty methodical. First, they take that specimen, your throat swab, or maybe another sample. Then they spread it on that same blood agar plate we talked about and let it grow. After that, a technician takes a look under the microscope, searching for those signature chains of round bacteria. And finally, for the ultimate confirmation, they do one last chemical test. And this right here is the aha moment. It's called the catalase test. It's simple. They mix a bit of the bacteria with a drop of hydrogen peroxide. Now, if it were something else, it might bubble up. But Streptococcus is catalase negative. That means no bubbles. And just like that, the case is closed. We've got our culprit. Okay, so we've identified the enemy, we've closed the case. Now how do we fight back? And the good news is, we have some really effective weapons in our arsenal. For decades, penicillin has been the go-to drug for treating strep infections. And for patients who have an allergy to penicillin, no problem. Doctors have great alternatives like erythromycin or cephalexin that work just as well. So we can identify this bug and we can treat it. But that brings up a huge question for the future, doesn't it? We have the tools to fight back now, but what happens when bacteria like this start to get smarter? When they learn to resist our best weapons? Well, that's a whole other case file and a story for another time. Thanks for digging into this with me.